So we've covered primary structure. The key thing to appreciate is some of the properties of the peptide bonds, the strength of the peptide bond. It is very difficult to break down. You need extreme acid conditions, harsh conditions to do that. Importantly, the sequence in that primary structure is what ultimately determines the function, structure and function of a protein or of a polypeptide. As I mentioned to you, many inherited diseases are based on a single change in the primary sequence. And I gave you the example of sickle cell anemia the other day. Okay, so very important thing to remember about the primary structure. So here you have a polypeptide. It's going to have some form, some organized or disorganized form in three-dimensional space. And that's really what secondary structure or the second level of organization of proteins is about. Secondary structure is about the three-dimensional pattern that a polypeptide can exist in. Okay? So there are three different common patterns, if you want, that create secondary structure. First one is called helical. Second one is beta. And the last one is called random chain because it doesn't have any specific pattern, but it still has a three-dimensional, non-repetitive structure. So these guys have repetitive structures and create a pattern, a helical pattern or a beta structure. And we will talk about both of them in quite some detail. Okay? So the first one, the alpha helical secondary structure, there are two types and designated by, here's the alpha helix. You can have a left-handed based on the direction of the polypeptide. In this case, it's sort of a left-handed helix or a right-handed helix going in the opposite direction. But this is the pattern, the helix, or that is formed, okay? That loops, those loops are repetitive and constant, okay? So how does this three-dimensional structure form and what stabilizes it? What keeps it in that pattern, in that helical structure? I just reminded you a few minutes ago the importance of the dipole moment of the peptide bond. Okay? Because that peptide bond can form hydrogen bonds. Okay? Because of the nature of that peptide bond. So I want you to look at this here. Here's a polypeptide, and that's the alpha helical structure. Here's one peptide bond between two amino acids in that polypeptide, forming a hydrogen bond, these dots there, with another peptide bond in that helix, within that helix. Actually, it so happens that these two peptide bonds are 4 residues apart, are separated by 4 amino acids. Okay? So, in principle, every peptide bond in an alpha helical structure is engaged in hydrogen bonding with another peptide bond in that polypeptide 4 residues away. Okay? And this is kind of demonstrated here. Without getting into all the details, here's one peptide bond interacting with another 4 residues away. Here's another one. Here's another one. Here's another one, and so on. Okay? We said a hydrogen bond is very weak, non-covalent. However, if you have 50 of them or 100 of them within that polypeptide, they're additive and they stabilize this alpha helical structure. Essential for maintaining the alpha helical structure. That's the only thing that is maintaining that structure, that secondary structure, are the peptide bonds between, the hydrogen bonds between peptide bonds within the polypeptide. You with me? Okay. Yeah? Good? All right. So here's again, 1, 2, 3. So this spans the entirety of the alpha helical structure. Now, what I want you to pay attention to is where the R groups are. They kind of fan out to the outside. So here's the alpha helix. And the R groups are fanning out to the outside. What's important about that is to appreciate, let's say this R group is polar. So let's say all of them are polar. So that's going to give that polypeptide polar properties. Maybe that polypeptide is able to associate with water and be water soluble. Okay? So very important to remember that. And this is really just another designation here. This is sort of an imaginary cylinder here through the alpha helix. There's not a cylinder like that, but it's to help you visualize the alpha helix. And again, and here are don't worry about any structures, but there are peptide bonds involved in hydrogen bonds with 4 residues apart. All of these dashed blue lines are hydrogen bonds between peptide bonds, creating this organized alpha helical structure. Okay? And the R groups in purple are to the outside of that axis of that alpha helix. So important thing is what maintains the alpha helical structure is the additive effect of the hydrogen bonds between peptide bonds 4 residues apart, 4 amino acid residues apart. There are amino acids that can disrupt the alpha helix. And those are proline and glycine. So that's important to know. If in the primary sequence, and I mean a polypeptide has a nice alpha helical structure, all of a sudden there's a proline in its sequence, that's going to disrupt that alpha helical structure. Frequently, proline and or glycine can bust. They call them alpha helix busters because they bust that. They don't break the peptide bonds, but they break the pattern, the alpha helical pattern. So these are a single proline or a single glycine. Other things that can disrupt are a large number of charged amino acids, but it has to be many. So let's say you'll have 4 or 5 aspartic glutamic acids in one part of the polypeptide in a sequence. Okay, they can disrupt the alpha helix. But there's got to be many of them, several of them. Or the third thing that can disrupt it is, again, many large, bulky amino acid side chains, like phenylalanine, tryptophan, tyrosine, and so on. But they all have to be, again, very next to one another. There's got to be several of them. One of them is not enough. There got to be 4, 5, 6 or more. However, a single proline or a single glycine can disrupt the alpha helical structure. Okay? This is just to kind of demonstrate to you and get you familiar with actually what proteins look like in the cell. This is not an artist's rendition, okay? This is an X-ray crystallography of myoglobin, and so that you won't get too scared by looking at it and say, what is that? Well, this is how myoglobin exists in the muscle. That's its final, in this case, tertiary structure, because it's folded upon itself. But I want you to appreciate that, for example, here you have one stretch of alpha helix, another stretch of alpha helix, another segment of alpha helix, a segment of alpha. All of these are alpha helices. There's actually 8 of them that are interrupted at some point. And they could be interrupted by the presence of a proline or a glycine or some other thing that interrupts the alpha helix. So this is the designation of an alpha helix, and I just want you to kind of be familiar with it. Okay. Okay, so you with me so far? Okay. The second type of secondary structure. Again, remember, this is about the three-dimensional configuration of the polypeptide in space, the three-dimensional spatial configuration. So the second type of secondary structure for polypeptides, for proteins, is called beta pleated sheet or beta sheet. First of all, for a beta sheet to form, you are going to need either 2 distinct polypeptides or 2 segments of the same polypeptide coming together. Okay? And they come together to form this structure designated by this wide arrow, where the beginning of the arrowhead is the N-terminal end, and the arrowhead itself is the C-terminus. That's the designation. So that's looking at the beta sheet from top. That's what it should kind of look like. Looking at it from the side, like to the profile of the beta pleated sheet, you'll see these pleats. There, 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 there. And again, it's the same designation, N to C. Okay, so this is what they look like. This is actually a pretty good representation, kind of like the how the accordion pleats are. You see here, here's one segment if you want, one polypeptide. Here's another one. And they form together, they form the beta and the pleated sheet. So basically, you can imagine this is almost like stretching those, and you create those pleats. So the second important question here to ask is, what keeps these two segments or two polypeptides? Either way, they can be segments from the same polypeptide or two distinct polypeptides. What keeps them together in this organized structure? The answer is hydrogen bonds. Again, in this case, this would be an illustration. So imagine this one of them. Here's another one. In this case, you look and see all the peptide bonds are forming hydrogen bonds with the opposing segment, with the peptide bonds of the opposing segments. So it's harder to see here, but there they are here, and so on. Okay? So again, importance of hydrogen bonds for maintaining the secondary structure of the beta pleated sheet. Now, we said that in order for the beta pleated sheet to form, you need two segments or two distinct polypeptides. The orientation of those polypeptides can be in the same direction, meaning, let's say, amino to carboxy, amino to carboxy. This would be called parallel beta pleated sheet. Or you can have the reverse orientation. Let's say amino to carboxy and carboxy to amino. So in this case, there will be anti-parallel. They would still form the same pleated sheet structure. As a matter of fact, this one here is anti-parallel because one of them is, this one is from amino to carboxy. The other one is from carboxy to amino. So the orientation is reversed, but they still form the beta pleated sheet. So they can be designated as parallel or anti-parallel. That's all. This is another kind of representation. You can have sometimes three polypeptides forming the beta pleated sheet. In this case, you have all oriented in the same way. This is the carboxylic end to the amino end. So these are parallel. And again, what's keeping those sheets together are these dashed blue lines, which are the hydrogen bonds. Okay? You can have, and these are called parallel, again, because all the polypeptides are oriented the same way, carboxy to amino. Or, if one of them is oriented in a different way, amino to carboxy, then this beta pleated sheet becomes anti-parallel. Okay, that's all we need to understand on that. Regardless of whether it's parallel or anti-parallel, what keeps a beta pleated sheet together are the hydrogen bonds between the peptide bonds of the opposing or adjacent segments of polypeptides. Okay? These hydrogen bonds here, what keep these beta pleated sheets together. Okay? So let's go to the next thing. You with me? Okay. So here's the structure there. Again, amino to carboxy. Here's a beta pleated sheet. This wide yellow arrow. And here's this blue short segment. There's usually this short segment is about 4 amino acids. In the 4 amino acids, you find either a proline or a glycine. One of these guys, if present, will create what is called this beta bend. That means you've disrupted the beta pleated sheet, and this is just a single row of 4 amino acids containing proline and or glycine. So you've disrupted the structure. Once after that, the beta pleated sheet can reform. Once it goes through this beta bend, it can reform, and now you have it going in the opposite direction, for example. And don't worry too much about these. These could be other non-covalent interactions between the R groups in the case of these two beta pleated sheets. But the important thing is a beta bend disrupts the beta structure and usually contains a proline or a glycine in this 4 amino acid segment. Okay? So we get to something called supersecondary structure. So supersecondary structure, the best way to describe it is a further organization of the secondary structure that creates another unique shape or pattern, but it's not quite tertiary structure. Okay? So it's intermediate between secondary and tertiary. And the easiest way, again, to kind of point out to it is usually supersecondary structure involves more than one type of secondary structure. So here, for example, here's an example of a polypeptide with supersecondary structure. It has a beta sheet, then it has an alpha helix, then it goes back to a beta sheet. So this level of organization is called beta alpha beta. You can have this type where you have alpha, then random, then alpha. So creating a further organized structure known as a supersecondary structure. There are many of them, and you don't need to memorize what they are, but you just want to understand that they're further organization of a secondary structure. So here you have a beta barrel. You see the beta sheets are in the form of a barrel. And here you have twisted beta sheets. And you have some other ones, beta, alpha, beta. All right, so I'm going to go back here just to tell you, you don't need to know the specific details, but understand what supersecondary structure. It's usually 2 or more secondary structures. And here's an example, beta, alpha, beta. Here's a more kind of organized one with called the Greek key, beta meander, and the beta barrel. So that's a specialized further organization of the secondary structure. Okay?